Our hypothesis for the first experiment was that the potato catalyst will cause the conversion from H2O2 to water and oxygen, which will result in the leaf chads floating faster.
Enzymes are proteins produced by cells, and they act as catalysts in biochemical reactions. They change the rate at which the reactions occur, so speeding up, slowing down, everything in between there. They're called a reduction reaction because it requires energy to activate the reaction.
Materials: hydrogen peroxide, paper chads, potato catalase, stopwatch, hole punchers, and tweezers and beakers.
Okay, procedure. Fill a 50 milliliter beaker with 30 milliliters of hydrogen peroxide. For a control, we put a chad in a beaker with hydrogen peroxide and no catalyst. In this experiment, the chads did not float.
For our second control, we dipped the chads in catalyst and then put them into a 50 milliliter beaker with 30 milliliters of water. These chads didn't float either.
For the real actual experiment, you first dip a leaf chad in the potato catalyst, and then place the chad at the bottom of the beaker with the hydrogen peroxide, and record how long it takes for the chad to float to the surface. Totally.
Yeah.
For the first experiment, the data that we collected said that the average time for a paper chad to rise up with the catalyst and the hydrogen peroxide was 0.924 seconds. And it didn't rise at all in either of the controls. So our data sort of says that it happens faster with the catalyst. Who would have guessed? For our second experiment, our hypothesis was that if we raised the pH of the enzyme of the potato catalyst, the reaction would take place slower. It would take longer for the chads of paper to rise. That was the hypothesis.
Materials: hydrogen peroxide, paper chads, potato catalase, stopwatch, hole punchers, and tweezers and beakers.
And bleach.
For the procedure, we slowly raised the pH of the catalyst by adding bleach to the catalyst in increments of 2.5 milliliters. After each increase in pH, we dipped a chad in the catalyst and then in the hydrogen peroxide. And we recorded how long it took for the chad to float. We tested each increment 5 times. The conclusion to our experiment is that as we increase the pH by adding bleach, the time that it took for the chads to float increased exponentially. The horizontal axis on the graph shows the bleach concentration as it increases, and the y-axis shows the time that it took for the paper chads to float.
So in conclusion, the higher the pH got, the longer it took for the leaf chads to float up.